DNA's full name is deoxyribonucleic acid. It consists of nucleotides and has a sugar phosphate backbone. It also contains a nitrogenous base. The nitrogenous bases consist of thymine, cytosine, adenine, and guanine. Adenine and thymine always pair together, while cytosine and guanine pair. It is formed in a double helix shape and resembles a twisted rope ladder. DNA replication depends on specific base pairing. It begins at specific replication forks on the double helix. The replication bubble expands. Then, enzymes called DNA helicase attach to DNA and separate the two DNA strands by breaking hydrogen bonds between the nitrogenous bases. This forms two separated strands. The free-floating nucleotides in the nucleus attach to the two strands according to base pairing rules. The enzyme DNA helicase links the nucleotides to form two new DNA strands. This creates two daughter strands, which are identical to the parental strands.
As the DNA is replicated, the cycle of transcription takes place. Transcription is the transfer of genetic information from DNA to RNA. It starts with a nucleotide sequence. Promoter DNA is the beginning of the segment and the terminator is the end. The RNA polymerase is the transcription enzyme made in the nucleolus of the cell. The messenger RNA encodes amino acid sequences and transcribes the message from DNA to ribosomes. A codon is a 3-nucleotide sequence in mRNA that specifies a particular amino acid or polypeptide termination signal. It's the basic unit of genetic code. For example, AUG provides the signal for the start of a polypeptide chain. UAA, UAG, and UGA are stop codons. RNA always has a nitrogenous base. These can be A, adenine, C, cytosine, G, guanine, or U, uracil. RNA always has a nitrogenous base. In RNA, these nitrogenous bases can be G, guanine, C, cytosine, U, uracil, and A, adenine. However, in DNA, these can be G, guanine, C, cytosine, T, thymine, and A, adenine. Messenger RNA is where DNA transcribes its genetic information to be carried to ribosomes for protein synthesis or translation machinery of the cell. In prokaryote transcription, RNA polymerase binds to a specific binding site known as the promoter. The promoter determines which of the two strands of the double helix will be the DNA template. During initiation, the polymerase attaches and starts RNA synthesis. Hydrogen bonds of the DNA are broken, and the DNA segment elongates as strands grow further apart. When the RNA polymerase reaches the section, base nucleotides pair with RNA nucleotides. The RNA elongates as synthesis continues. RNA peels away from its DNA template, allowing DNA strands to come back together at the transcribed regions. Then the polymerase reaches the terminator. The molecule detaches from the RNA molecule and the DNA. Eukaryote transcription occurs in the nucleus. It is similar to prokaryotic transcription but with a few modifications, like the addition of extra nucleotides to the ends of the RNA transcript, such as a small cap made of guanine,
Point to the tail.
and a long tail which is made of a chain of 50 to 250 adenines. The cap and the tail facilitate the export of the mRNA from the nucleus, protect the mRNA from attack by cellular enzymes, and help ribosomes bind to the mRNA. The cap and tail are not translated into protein. In most protein-coding genes, the DNA sequence that codes for the polypeptides is not continuous. Most plant and animal genes have non-coding regions called introns. The coding regions are called exons. Both are transcribed from DNA to RNA. Introns are then removed and exons spliced together, known as RNA splicing. RNA splicing is begun by a complex series of proteins and small RNA molecules in most cases, but RNA can catalyze the process itself, and RNA can act like an enzyme. An Ozaki fragment is a relatively short fragment of DNA created on a lagging strand during DNA replication. When the lagging strand is being replicated on the original strand, the 5 by 3' pattern must be used. Thus, a small discontinuity occurs and an Ozaki fragment forms. These fragments are processed by the replication machinery to produce a continuous strand of DNA and hence a complete daughter DNA helix.
A ribosome consists of two subunits, each made up of proteins and a kind of RNA referred to as ribosomal RNA. First, an mRNA molecule binds to a small ribosomal subunit. A special initiator tRNA binds to the specific codon, called the start codon, where translation is to begin on the mRNA molecule. The initiator tRNA carries the amino acid methionine, abbreviated MET, and its anticodon UAC binds to the start codon AUG. Next, a large ribosomal unit binds to the small one, creating a functional ribosome. The initiator tRNA fits into one of the two tRNA binding sites on the ribosome. This site, called the P site, holds the growing polypeptide. The other tRNA binding site, called the A site, is vacant and ready for the next amino acid